And welcome to the webinar. This is a National Association of Plant Breeders webinar. It is, we organize these sessions almost on a monthly basis during the fall and winter seasons, and we try to bring in individuals and professionals from all over the industry and academia to bring in different perspectives on technical skills as well as emerging technologies, and sometimes just overall general discussion on the on technology, emerging technology and existing technology in plant breeding. So we're very happy to have you here today and to get the opportunity to share some of the work that we do in our lab, which involves using AlphaSimR. So what I was asking earlier was that before we begin the workshop, it would be best for, since we're going to be here for a couple of hours running the workshop, I usually try to start these things with some sort of idea or sense of what is everybody expecting, or what would you like to get out at the end of the workshop. So by the end of the workshop, what is the objective that you feel you want to achieve? So you can either type it in the chat or feel free to unmute yourselves, and we can spend a few minutes at the beginning here having that discussion.
A direct message to me came through saying basic understanding of how to use AlphaSimR such that I can apply it to a wheat breeding program. In particular, I'm interested in balancing early generation yield testing versus selfing/purifying time.
Wonderful.
That's great.
That's a great one. And I apologize if we forgot to introduce ourselves. My name is Valerio Hoyos-Villegas. I am I'm an assistant professor in the Department of Plant Science at McGill University, and the lead of the pulse breeding and genetics laboratory here at McGill. Our lab is based in Montreal, Canada. Isabella, you want to go ahead?
Sure. Hi, everybody. Thanks for being here. I'm a second-year PhD student in Valerio's lab, and I've been doing a lot of simulation work using AlphaSimR as the platform for my research. I have an undergraduate degree in sustainable agriculture from the University of Massachusetts.
Wonderful. Okay, we're just carrying on with the discussion. Here we got another one from Shayla. Hello, just wanted to learn more about genetic gain plus how AlphaSimR works. Well, that's we hope that that we accomplish that today. I think we share that goal with you. Any other thoughts? And Dean, Dean is as IITA. Hello, Dean. Cheers from Nigeria. I want to learn how AlphaSim works and how it can be used to simulate genetic gain. Okay, a lot of interest in genetic gain, and I see. Well, that's certainly a part of the simulation, right, Isabella?
Yeah, we're going
to look at today.
Yeah, absolutely. That's really cool.
Mm-hmm. Okay. Nanette, working forest genomics, want to simulate breeding populations with a range of genetic and experimental design parameters to see the effect on GS accuracy. Wonderful, that's fantastic. Yes, we will look at that. We have a lot of interest in that as well, and that's part of our workshop today here. And forest genomics, I guess it's very difficult to generate populations to validate these types of things. So I'm sure you find a lot of use from these types of approaches. Edwin, looking for selection F2, F3, F4 generations specifically for trees. Okay, and another tree person. Fabian, learn what sort of questions that AlphaSimR can and can't address if I can't use it to complement my research in GS sorghum breeding program at Texas A&M. That's an interesting thought. I mean, there's certainly AlphaSimR has a lot of options. In my lab, we've used in the past, we've used QLine Plus. I've actually been part of the development team of QLine Plus of Gene suite with the University of Queensland in the past. And there are certainly things that one can do that the other one can't, and vice versa. So it really depends on what sort of question you want to ask. But what do you think, Isabella?
Yeah, I think that this platform is extremely flexible, and hopefully you'll get a sense of that today. But it's built to be flexible. So I think there are few things that you're going to find that you can't address. In today's workshop, you might not see exactly what it is you're looking for, but there's extensive documentation on what AlphaSimR can do, and I can certainly send that to everybody as a resource.
Wonderful. We still got people coming in. We had quite a bit of interest in this workshop, so I just want to give a little bit of time, because we still see people joining. But this is very exciting to see this kinds of ideas popping up here in the chat. So if you have any others, feel free to type them in. One that we did with a previous student of mine was that we wanted to find out exactly how hard it would be to accumulate all of the QTL that were known, that were reported for yield and white mold tolerance in a single line. So what would be the exact probability that we could recover a single line from a breeding program if we were to incorporate all of the QTL that have a favorable effect on yield in common bean. So that was quite interesting. We did a number of approaches using number of breeding strategies, as well as multiple breeding frameworks, as we call them, like genomic selection, conventional, and speed breeding, and at different parental levels, different number of initial parents. And we found that if you have a very, as might be expected, you have a very small number of initial parents, it can be quite hard. But what was quite interesting was that genomic selection didn't prove to have an advantage when it came to very small breeding programs. So that was a bit of an interesting learning from that. All right, we'll go just a couple more minutes. Any other thoughts? Shayla, so great idea. Just wondering how long it will take to add these QTL into one single line, and what about linkage drag along with it? Yeah, that is a great question. And linkage drag, you can certainly simulate linkage drag. I don't know how you would. I think you could potentially simulate linkage drag in AlphaSimR, Isabella.
Yeah, there is a framework for it. It's not something that I personally have a ton of experience with yet, but it's in there.
Yeah, you can certainly do it in QGene. Well, what you do is that you link two loci, and then you assign a negative effect to one. And so basically when one is inherited, the other one is linked to it, and it drags down the effect. And so that's sort of like simulating linkage drag. You can certainly do that. I'm sure that you could come up with a clever way of doing that in AlphaSimR too, if it's not directly coded as a function. All right, I think, what do you say? We get going?
Let's get started. Yeah.
Wonderful. All right, go ahead, Isabella. It's all you.
All right, thank you. All right, everyone. So thanks again for being here in this workshop today. I'm going to share my screen. We're going to be starting with a brief introductory presentation. And then after that, we're going to spend a large chunk of the workshop in RStudio Cloud Workspace, exploring some scripts in AlphaSimR. So let me present here. Okay, I hope everyone can see my slides. So we're going to be exploring breeding program simulation using AlphaSimR today, as you all know. So brief overview, we are going to start with an introduction. I'll tell you a bit about what goes on under the hood, so to speak, in AlphaSimR. And then we're going to be using AlphaSimR to explore the breeder's equation and kind of vice versa, use the breeder's equation as a framework for exploring some of the basic functionality of AlphaSimR. So AlphaSimR is a software package that we can use to simulate plant and animal breeding scenarios. It was developed at the University of Edinburgh, and it makes use of R commands for easy implementation. This package is very, very flexible. You can tailor it to meet your needs and your interests. And something else that's really great about it is that you can use it seamlessly alongside other R functionalities or other R packages if you have some knowledge on how to do basic coding in R. So we're not doing this today. This is just for your future reference. If you want to use AlphaSimR on your own for your own purposes, you're going to have to download R onto your device. And I also recommend R Studio just to make things a little bit more user-friendly. And then once you're in the console, you just have to install the packages and load in the library, and then you're ready to go. There is a reference manual available that outlines all the commands in AlphaSimR. So in this workshop, we're not going to cover everything AlphaSimR can do. It's very versatile and very extensive. So if there's something you want to do that you don't see today, I'm sure that you can find it in the reference manual, and I can send that to everybody. Yeah, and I see, Valerio, you said don't hesitate to ask questions. Please unmute yourself, pop in the chat if you have any questions. Okay, so how does AlphaSimR simulate a breeding population? When we create a population, there will be an object in R that contains all of your individuals and all of your genotypes, as you can see here in this data frame. So we have a row for every individual and the genotype at every locus that we define. Genetic and phenotypic values will be distributed around desired mean set by the user. And it uses Markovian coalescent simulator command to generate realistic biallelic sequences. So it basically just constructs a demographic history. This is sort of a backwards in time simulator. AlphaSimR also has some predefined parameters for certain species. So if you're working on cattle, wheat, or maize, those demographic histories are preloaded, but you can also define your own parameters if you are working with a different species. AlphaSimR also models meiotic recombination and linkage, which we mentioned briefly in our discussion earlier. And we can also define biological effects for our traits. So AlphaSimR can model additive, dominance, epistatic, or genotype by environment effects. Within those dominance effects, you can also have variable degrees of dominance, and we can define different combinations of these effects seen here. You can do additivity and dominance, additivity and epistasis, etc. There's also commands that allow you to define a custom selection scheme. So today we're going to be coding a simulation of the pedigree method. So what I recommend you do if you want to simulate a certain scenario is write it out and then find the corresponding command. So in our pipeline, we'll start with some random crosses of selected parents. So we'd use this command randcross. And most of these commands in AlphaSimR take several arguments. So when we rand cross, we're going to define the population, the number of crosses we want to make, and the number of progeny per cross. Then we self, so we can use the self command and define the population and number of progeny per selfing. Then in the next generation, we'd go on to select within families. So we would use the select within fam command, define the population we're selecting within, how many individuals per family, and the trait we want to select on. When we go to make selections, there's this argument called use equals. So you can tell AlphaSimR, I want to use phenotype to make selections. I want to use genetic values. Or if we're working with genomic selection, you could say, I want to use the estimated breeding value to make selections. So you have a lot of flexibility there. I won't go through every single line here, but I also want to point out down at the bottom, if you want to simulate yield trials, you can indicate that you want to phenotype several reps. So again, this is the pipeline we're going to be simulating today, but you can use these commands to simulate whatever works for your needs. All right, so hopefully we've all seen the breeder's equation. A couple of people mentioned they want to learn about genetic gain in today's workshop. So the rate of genetic gain is defined by selection intensity, selection accuracy, our additive genetic standard deviation, and the length of our cycle, our breeding cycle. So if we want to increase the rate of genetic gain in our breeding program, we can work with these different variables. And that's what we're going to be doing today. So we're going to, in a few minutes, head into our Studio Cloud workspace, and we're going to run 5 simulations. And we're going to be evaluating genetic gain and genetic variance after one breeding cycle for each of these simulations. So we're going to start with just a baseline phenotypic selection pipeline following the pedigree method. And then we're going to do a little bit of single variable manipulation to see how that impacts our genetic gain and variance. So we'll start by first altering our selection intensity. Then we're going to change our selection accuracy. Then we'll shorten our cycle time. And our final script, we're going to see how we can implement genomic selection. A couple people in the chat earlier mentioned they were curious about GS, and we will look at that today at the end of the workshop. So at this point, I'd like to head to our workspace, but before we do that, are there any questions? Okay, sounds like no questions. So what I'm going to do is pull up the RStudio Cloud workspace in my Zoom screen share, but I encourage everybody to sort of minimize Zoom, just listen and focus on the RStudio Cloud workspace on your own. Everyone should have received a link to that workspace yesterday. And if you don't have the workspace, I can send a link in the chat. So at this point, everybody should be going to the AlphaSimR workshop, Posit Cloud, RStudio workspace.
Maybe thumbs up if everybody's there or when everybody gets there.
Yeah, that'd be great. Thumbs up when you're in your workspace. Okay, I see a couple of thumbs up. That's great. We'll wait to see a few more thumbs up before we move forward.
That seems to be everybody, I think.
Okay, great. Awesome, thank you. Cool, so,
Or somebody got something in the chat.
so your console should be empty. but the workspace should contain several scripts that will be opening one at a time. So I'll give you a little background on what this workspace does and how it's set up in case anybody's new to using RStudio. So there's kind of 4 boxes here. The top left is where our scripts are going to show up. The bottom left is our console. That's where all of our commands are going to run. The top right, we have our environment, and that environment should be empty, and we'll fill it up as we go. And that environment is going to contain all of the objects and variables and things that we define. And the bottom right should have a list of 7 scripts. So first thing we should all do is open up script 1. It's called simparams. If you click on it, it should open a file in the top left box. So can we do a thumbs up once everybody's in script 1 called simparams? Okay, see a couple of thumbs up. That's great. Okay, I think I'll go ahead. So script 1 is the script where we're going to define all the parameters of our simulation. So this is something that you do at the beginning of your simulations to kind of create this global environment that you're trying to simulate. These are all R Markdown files. If you've not used Markdown before, the scripts, the code that we want to run is in these gray chunks. And if you hit the little green play button at the top right of the chunk, it's going to run the code down in the console for you. So this first chunk, load and install required packages, you can press play. If you get a pop-up, you can hit no. You might not get a pop-up, but I did. And then down in the console, it'll install the packages and load the libraries. Okay. So for each of these scripts, I'm going to just walk us through each of these chunks. So chunk 1 in our sim params is going to be to establish your founder population. So line 17 of this script, we're going to define the number of individuals in our population, in our founder population. So in front of those pound signs where it says n int equals, I'd like everyone to type in how many individuals you'd like to have in your population. I put maximum 500 here. You can do any number theoretically, but for the sake of computational time, we're going to set some maximums for today because it will take longer to run if you have a gigantic population. So we're going to define number of individuals, number of chromosomes, and the number of segregating sites that's per chromosome. Then in line 21 of this script is the syntax you'd use to define your founder population. So it says founder pop equals, and then run max is the AlphaSimR command that is going to allow us to create that biologically realistic population. And we've already defined these variables for individuals, chromosomes, and seg sites. So we can run that chunk. And depending on how big your population is and how many segregating sites you have, it's probably going to take a few moments for that to finish. Can I get a thumbs up if you have your founder pop? Cool, see a few thumbs up, that's great. All right, so next chunk, we're going to be defining our trait. So first thing we're going to do is determine how many loci we want controlling that trait. Beware that this number needs to be smaller than the number of segregating sites. So for example, I said 100 segregating sites per chromosome, and I'm going to say I want 5 loci per chromosome controlling this trait. You can set the mean of your trait and the heritability. Later on, we're going to be increasing our heritability, so I encourage you guys to put a heritability 0.5 or less here. Somebody asked, is it possible to create a founding pop from SNP datasets of actual breeding lines? The answer is yes. I've just done that myself. There's a little bit of work on the front of that to put your SNP data into a format that's readable by AlphaSimR. But that's totally possible. Yes, you can use real data. That's a good question. Yes, so if you want to use real data, you're going to have to provide SNPs and a linkage map, and you're going to need to like separate your SNPs into per chromosome so that it'll line up with your linkage map. If anybody wants to do that and runs into any trouble, I have a script that will turn SNP data into files that are readable by AlphaSimR, and I can share that in the future if people need. Okay, great questions. So back to our scripts. Once you define your trait, we can play this chunk. And in line 30,
you got a question, Isabella? Is it possible to create funding pop from SNP datasets of an actual factual breeding lines?
Yeah. So that's totally possible. That's something that I've done. You just need your SNP data. You need your SNP data and a linkage map, as somebody mentioned also in the chat. And yeah, there's not a ton of detail on how to do that. It took me some time to figure it out, but to the person that wants more information on that, maybe you can reach out to me and I can share a script with you on turning real population SNP data into an AlphaSimR readable file. Yeah.
Second question from Prashant. I couldn't find too much documentation about it. Can you point me in the right direction? So perhaps something we could probably put together an email as an exit email for the workshop, and we'll send you some links.
Definitely. Yes.
Yep.
Okay. Another question. Okay, cool. Great. Okay. So a really important step in your simulation is going to be what happens here in lines 36 to 38. So we're creating an object called SP, stands for SimParams, and we're going to create a new founder pop. Then we're going to add a trait. So it says in line 37, SP $add trait A. So here, if you wanted to do a trait that was just dominant or showed additivity, epistasis, and G by E, you can change it to say add trait AD or add trait AEG to represent those different effects. It's up to you what you put there. So once you have defined your trait, what I'd like everyone to do is go down to your console and you can type founder pop into the console. And it should give you some information about the object that you just created. So it's an object of class map pop. It'll give you the ploidy, the size of the population, and how many chromosomes. And loci are there. And somebody mentioned their posit is blank. Is anybody else having a blank environment for their posit? So you can definitely follow the Zoom, but I'm going to share a link in the chat to the environment. You can try that and see if it'll give you the scripts. Try that. Okay, so where are we at? So right now I'm at chunk 4. It's line 48 of script 1. And here we're going to just look at the genotypes. So we're using a command called pull segsite geno from our founder pop. And then in line 52, we're just taking the first 5 rows and first 10 columns of that data frame. And if you play that chunk of code, it'll show you the genotype of each individual at each locus. So the names of these columns where it says 1_1, 1_2, that just means first locus on the first chromosome, second locus on the first chromosome, etc. We can also pull individual chromosome data. So the next chunk on line 57 similar command, except it's showing you each individual chromosome, both of them for our diploid individuals. And then chunk 5, we can view some genetic values and phenotypes. And this is just to give you an idea of what we've created here. So if you run chunk 5, you'll get some values down at the bottom. The first values are the genetic values up at the top, and then the second row are your phenotypic values. And you'll see that these values are similar, but they differ slightly. They're going to be distributed around the mean that you defined earlier. The genetic values are basically a summary of the marker effects, and the phenotypic values will include an adjustment for that heritability that we set. So I hope that this script gave you a sense of the basics of how to set up a simulation and what we actually create when we define these parameters. So we'll move on to the second script. I'd like a thumbs up if you're ready to move on to the next script. Cool, awesome. So script 2 is going to be where we establish our baseline pedigree pipeline. And we're going to be defining some values throughout the workshop so we can compare the performance of these different scenarios. So this first chunk, I called it chunk 0. We're going to just create some empty matrices to hold the results of our simulations. So you can play that chunk, and we're not going to really worry about it now. We'll come back to those matrices later. But we're going to be looking at genetic gain values and variance values for our breeding cycle. Okay, chunk 1. This is where we're going to start our breeding cycle. So line 20, I'd like everyone to define how many crosses you want to start with. I put maximum 25. Again, that's just for like computational reasons for the sake of our workshop. You could do as many crosses as you want. And then in line 21, you're going to define the number of progeny per cross. And I put a maximum 10 there. And then once you have defined number of crosses and number of progeny, you can play this chunk of code. And I'll tell you a little bit more about exactly what's going on here. So line 23, we created a new population of parents from our founder pot. Then in line 24, I said F1 is going to be equal to the results of our random crosses from our parent population. And then we define some arguments, number of crosses and number of progeny per cross. There is the option to do a specific crossing scheme, if that's something you're interested in. For the sake of simplicity, we're going to just do random crossing today, but you could create a crossing design if that's something you'd like. Lines 25 and 26 of this script, it's just putting some data into those empty matrices we created. So we're going to be looking at the number of individuals, n-ind, and the mean genetic value for each generation. Then line 30 and 31, we're creating our F2 by selfing our F1s. And in the next chunk, that's when we're going to start making our selections. So I'll tell you a bit about what we're doing here. There's nothing for folks to type in at this point, but line 43, we are selecting within family. So in the pedigree method at the F2, we'd make within family selections. We are defining the number of individuals we want to select from each family. And we're going to use the phenotype to make those selections.
Isabella, you got a question? In potato, we use 200 progeny per cross. What happens in this case?
So that is something you could definitely do. The thing is, it might just take a while. Like the command where you are generating those progeny might take like a couple of minutes to complete, but you could still totally simulate that. Hope that answers your question. Okay. So chunk 2 of our script just runs through the pedigree breeding pipeline, where we're selecting within family, and then we select families, and eventually we select a single line to serve as our variety. So people can play that chunk. It'll run through our pipeline. So this is essentially one breeding cycle that we've just simulated. And the next thing we'll do is we'll make some visuals for what we just did, because it can be a bit abstract. So chunk 5, we're going to graph how the size of our population changed over our breeding cycle. So we've just pulled the number of individuals for every generation, and then we'll plot them. So if you play chunk 5, you should get a graph that says breeding population size for one cycle. And we can see that we start with our founder pop at the F2. We've really kind of amplified our population. And then after the F2, when we start making selections, the size of our population kind of rapidly declines to the end of the breeding cycle. In chunk 6, we'll visualize how the genetic values have changed. So we pulled out the genetic value, mean genetic value for each generation, and we'll plot those. And hopefully what you'll see is it kind of just continually increases over the course of our cycle. So what we've done is we've outlined a breeding pipeline where the number of individuals will decrease as we make our selections, and the genetic value will increase as we make our selections. Any questions so far on the pipeline that we simulated? I'm going to assume no, I don't see any questions. So chunk 7. Here we are going to pull the genetic values and the genetic variance, and we're going to put those values into a data frame. So the last thing I want us to do in this script is you can go down to the console and type in results with a capital R. That's the name of our data frame. And you should see 2 values for genetic gain and delta variance under baseline. So our baseline genetic gain will be a certain number of units. It's going to be different for everybody. Even if we all had assigned the same exact values, we'd get slightly different results because it's a stochastic simulation. But you can see how much the variance changed and how much genetic gain we achieved. Okay, so that's it for script 2. If folks want to give me a thumbs up, we'll move on to script 3, where we're going to increase selection accuracy. Cool. Awesome. Thank you for the thumbs up. So script 3, increase accuracy. This script is going to increase the accuracy of our breeding program by increasing the heritability, the narrow sense heritability. And the idea behind this is that we can define our accuracy as a correlation between the breeding values and the phenotypes that we have. And because additive variance, which defines our narrow sense heritability, is the portion of our genome, the genetics that gets passed on to the next generation, we can sort of equate the breeding value and the narrow sense heritability. So the square root of heritability is going to be equal to essentially our accuracy. So they're proportional. So theoretically, if we increase the heritability in our population, we should be increasing our accuracy and maybe increasing our rate of genetic gain. So with that in mind, we're going to redefine our simulation parameters. In line 10, we're going to define a new variable called H2 updated. And here I like everyone to type in a higher accuracy than the one you did previously. So for me, the baseline heritability was 0.5, and now my heritability is going to be 0.9. And we can run that chunk to redefine our parameters. And the rest of this script is identical to the baseline. We're just doing the same breeding pipeline with a higher heritability. So you can go ahead and just play all those chunks. Chunk 2, chunk 3, and then you can type results in to view your results. For me, I actually had a decrease in genetic gain here. I don't know if other people saw the same, but interesting. Yeah, so script 3, pretty quick. Hopefully you all have some new values in our results data frame, and we can go ahead and move right along to script 4. Okay, so script 4, we're going to move on to the next variable in our breeder's equation, which is selection intensity. So this script is going to increase selection intensity in our breeding program. If we increase the size of the starting pool of selection candidates, but keep the number of selected the same, we're effectively increasing intensity. So the first chunk here, we're just resetting the parameters back to our baseline. So go ahead and play that. And then the next chunk, we're going to increase the number of candidates, but hold the proportions selected the same. So line 15 for n crosses updated, I want everyone to double the number of crosses that you started with. So here you can type n crosses times 2. And then play that chunk to redefine that variable. And then we will run through our pipeline again by playing a chunk 2 and chunk 3. And then we can view our results. So you can compare genetic gain for your baseline pipeline, and then the genetic gain for what happened when we increase the accuracy and increase the intensity. We can also look at how large the decrease in variance was over the course of our pipeline. Okay, let's pause for a second and please give me a thumbs up if you have run through script 4 with no problems. Nice, awesome.
Yes, when you do it, I don't know if it's possible. I want to go to the summary, right? I want to make use of your article. Thank you.
I couldn't quite hear what you said. Maybe type in the chat. Yeah, okay, if somebody has a question, I'm happy to answer it via the chat. But for now, let's go to script 5. So here, we're going to be implementing rapid cycling. We're going to kind of create a population improvement framework in our breeding program. And we're going to be selecting our parents from this improved pool. So just for good measure, again, we're going to set our baseline simulation parameters in the first chunk. We'll bring our pipeline through to the F2 in chunk 1. And here we're going to be recycling our F2s back into our crossing block to kind of elevate the mean of our population before making our new parent selections. So I'd like everyone to decide how many rounds of cycling you'd like to do here in line 25. And then in chunk 3, we're going to be running these cycles. So this is one of those cases where if you have a little bit experience coding, you can use an ER command alongside AlphaSimR command. So this is a for loop here. We're telling R that we want to loop over the code a defined number of times. So I set my number of cycles to 8, and it's going to basically line 35, select new parents from the F2. We're going to be using phenotype to make those selections. And where it says top equals true, that just means we want to be selecting the top individuals as our new parents. Then 36, we have our F1 from rapid cycling. We're going to randomly cross those parents. And then in line 37, we're going to self them to create F2, and then it'll cycle those F2s back as new parents for the number of times that you defined. And this is a step that might take a few moments because it's running the code over and over. It won't be as fast as some of those other chunks. Okay, so now we're going to go through our pipeline with those new parents. So line 46, I define this variable parents rapid. We're going to select individuals from the latest F2 population, which has gone through those multiple cycles. And then from those new parents, we will run our pipeline. I got a warning in my script, which you might have as well, but that's okay. And then again, we'll calculate genetic gain and the change in variance. And I'd like everyone to type results in to view the results. So for me, looking at genetic gain, so far the rapid cycle simulation has created the greatest gain. And actually the smallest decrease in variance. So that's interesting. Is everyone all set on script 5? Can we thumbs up when we're ready to move on? Awesome. Thanks, everybody. That's great. Okay, so in this script here, we're going to be implementing genomic selection. This script is the most involved, so it should take us a bit more time to run through than the others. But the idea is that we are going to train a machine learning model to predict the the estimated breeding value of our selection candidates. And GS, in theory, should increase our selection accuracy if we're making good predictions of breeding values. And this will also get at the cycle length variable in our breeder's equation. Because if we're not spending the time phenotyping, we're only going to genotype to make selections. The gain that we get from this simulation should be for a smaller unit time. So our gain per unit time with genomic selection is ideally larger. When we implement genomic selection, we first need to develop a population that we can use to train our prediction model. So if you're not super familiar with genomic selection, what we do is we train a prediction model using genotypes and phenotypes. So the model is going to be learning how to use the genotypes to predict the breeding value of a selection candidate. And then we'll use that model to predict new candidates and estimate the breeding values based on marker data. So chunk 1 is where we're going to develop a training population. So this is just a simple selection scheme where in line 10, we start with a random cross of our new parents and we'll self. In line 13, we're going to select within family at the F2. And then at line 19, we're going to select within family at the F3. And then we'll select top families. So we can play that chunk, and we've created a population that has hopefully some stable marker data. And in chunk 2, we're going to be defining our training population. And for this training population, we're going to use both our parents and our PYT data just to create kind of like a large, diverse training population. So line 33, we have this AlphaSimR command called pheno. That is where we pull the phenotypes. We're doing that in line 33 and line 34. And then line 36 and 37, we're using this command called pull segsite geno, and that is going to give us our marker data. And then in line 39, I'm just creating one large data frame with all of our phenotype data and all of our genotype data. So let's play that chunk. And after you run chunk 2, I'd like you to go down to the console and type nrow training pheno. Or you can just copy paste from the R script, and that will give you the number of observations that you have in your training population, just for curiosity's sake, how many individuals we're training our model on. And that's going to vary person to person depending on, actually, no, it should be 250. We should all have the same one for this one. So chunk 3. We want to be able to look at how our model performs when we're estimating our breeding values. So chunk 3 is just an empty matrix where we're going to add in model performance values. And we're going to define model performance as a correlation between the true breeding value and the estimated breeding value from our model predictions. Chunk 4 is where we're actually going to build our model. So something great about AlphaSimR is it has a built-in function for running the RR-BLUT model, which is kind of the go-to genomic prediction model that most people will use. But here I want to demonstrate how you can use external programs within AlphaSimR. So we're going to build a machine learning model called a support vector machine. We don't have to go into the specifics of how this model works, but I chose it because it's pretty quick and easy to implement. So line 63 of our script, we're defining a variable called training, and that is just all the genotypes and phenotypes that we're going to use to train our model. And then in line 64, we are just naming the columns in our data frame because the model is going to use those for reference. Okay, and then line 66 is where we are actually building our model. So we're going to call our model predict GEBV. And then we're using this command called SVM. This is not an AlphaSimR command. This is from an external package that we loaded earlier. And what this is going to do is train our model to predict phenotypes, which I defined as ID1 in the previous line of script, using all marker data. So you can play that. Building the model might take a few moments. For me, it was pretty quick, but I'll give a second for that to run for everybody. Okay. And then chunk 5, we're going to run our pipeline using our estimated breeding values as opposed to phenotypes, which we've been doing throughout all of our other scripts. So as always, line 75, we're going to start by creating a new founder pop of parents. Then in line 77, in order to implement genomic selection in AlphaSimR, we're going to pull all of our marker data. So we're going to use that pull segsite geno command again on our parents. That is going to create a data frame of genotypes, and we need to name all of the columns to correspond with our model, which is what's going on there in line 78. And then line 79 is where we actually make our predictions. So we're defining a variable called EBV of parents. We are using a command called predict. And then it says, so it says predict, and then in parentheses, predict GEBV. So that's telling we're going to make a prediction using our predict GEBV model on the genotypes of our parents. Okay, and then line 80, this is an AlphaSimR command. We say parent at GEBV. That is going to add a feature to our parent population of GEBVs. So we're setting our model predictions as the EBVs of our parents. Moving on to the next line of code, line 82. We're going to determine how well our model performs. So model performance 1 is going to be the correlation between the true values of our parents and the estimated values. And then in line 85, we're going to select some new parents for our next breeding cycle. And this is the same command that we use in our other scripts, select individuals from our parents. We're going to use n crosses as how many parents we want. The thing that's different is we're using the command use equals EBV. So we just set our estimated breeding values, and now we can use those to make our selections. And then line 89 onward is, again, the same pipeline that we've been running, except we're using estimated breeding values to make our selections. So if you haven't already, you can play chunk 5. And chunk 5 is going to bring us to the F5 generation. Something to keep in mind with genomic selection is that our model is not going to do so great as it kind of moves in distance away from the training population. So at this point, we're going to update our model. We can call it a model retrain. So lines 145 and 146, I'm just removing our old training population data, and we'll pull the F5 phenotypes and genotypes to build a fresh model for the rest of our pipeline. So you can play chunk 6. It's the same as our original model build, except we're using new data. And then chunk 7, we're going to continue down our pipeline.
Isabella, maybe worthwhile asking anybody if they are not at this point, if they're behind.
Sure.
Just give everybody a little bit of a break to see if you are behind. Just flag and we'll try to sort it out.
Yes.
There's a question from Nelson. Why have you estimated accuracy as a correlation of genetic value and EBV? Why not a correlation of phenotypes versus EBV? It's a good question.
That is a good question. We pulled EBVs here because GVs here because we've been using GVs for our other scripts. So in a sense, we are using genetic value here just for consistency for our simulations. But in general, when we're talking about genomic selection, we're not necessarily predicting phenotypes. We're predicting breeding value. So we wouldn't necessarily want to compare phenotypes to the EBVs. I hope that answers your question. I don't know if you want to add anything, Valerio.
No, I think that's fairly accurate. So the way we conceive this accuracy is based on the idea that the genotypic value the EBV, the GBV is a direct estimate. It's under the assumption that the GBV, or what you mentioned as the EBV, is a direct correlation with the genotypic value. Okay, let's bear in mind that the genotypic value is not something that is readily estimable, right? But we can approximate it via the use of markers. If we were to say have a way of if we didn't have markers, right, and we just wanted to estimate our accuracy as a mean of our ability to make selections using phenotypic data, then it would be a case of what you're mentioning in that second situation where it's phenotypes versus EBV. But you would have to have some estimate of the breeding value via other means, if that makes sense.
Yeah, definitely.
Jean-Marc, the SMV function calls a script that uses RBLUP.
The SMV function. So we're not yet. The SMV and RBLUP are exclusive. I mentioned RBLUP earlier because AlphaSimR has an RBLUP function within it, but this is totally external. I'm not sure if that answers your question. Okay, cool. Yeah, that's a good question. Yeah, if you are interested in using RBLUP, it's easy to implement in AlphaSim, and there's very straightforward instructions on that in the reference manual for the package. Any other questions? Those are really good questions. While we're paused, Valerio, I expected a lot more questions and technical difficulties, so we've run through all the scripts very fast. We maybe have some questions at the end, but I've created a space for people to kind of play around with some commands at the end if they want to do that. I'm not sure what your thoughts are.
I think that's great. Yeah, it seems that we're running smoothly.
Yeah.
So yeah, I think that will be okay. I mean, I'm just being a little bit weary or cautious with that anybody's behind, but it doesn't seem to be the case.
Yeah.
Nelson asks, will you also demonstrate how to simulate several traits using a selection index?
Yes, that's a really good question. I wonder if maybe we could finish getting through this script before we get into another topic, because we're almost done. And then we can talk about that question. Does that sound good? Okay, awesome.
So as I see things, it seems that nobody's behind, so I think we're great.
Yeah, you guys are doing a great job. So where I'm at here is I just ran chunk 7, which is continuing down our pipeline. And the script will print out a little data frame of our model performance. So it'll show you for each generation how well the model made its predictions. Again, correlation between the GVs and the EBVs. And we're all going to have slightly different values, but hopefully this will give you some sense of how well the support vector machine did. SVM is not typically used for genomic prediction. It's not super accurate compared to RBLUP, but it still works. So we can just finish the end here. Calculate genetic gain and change in variance for the cycle in chunk 7. We can type results to see how we did. Me personally, the GS genetic gain wasn't amazing. It wasn't as high as the rapid cycle. But keep in mind with GS that if we're talking about gain per unit time, GS is theoretically faster. So that's just something else to keep in mind. The last thing I'd like us to do to wrap up all of these simulations is visualize our results. So this is, again, a case where bar graph, making a bar plot is like a basic R command that you can use alongside AlphaSim. So if you run chunk 8, it's going to show you the genetic gain in units for each of these different scenarios that we ran. So if you're using AlphaSimR to run some experiments and you want to provide a visual to show how your experiments went, you can easily do that. And chunk 9 is going to show delta variance. So this is a decrease in variance. All these values are going to be negative, but you can see which variable had the greatest impact on reducing genetic variance over the course of our breeding cycle. Okay. Any questions? Those are all the scripts I prepared for you guys. I went really, really smoothly, so we're very ahead of time. But if there's not any questions on that, we can talk about simulating several traits. So I think if you want to do multiple traits, you want to define those traits in your sim params. So I can show you this in my screen share, if you guys want to return to the screen share. Can everyone see my screen? Thumbs up, you can see the screen. I'm in.
Yep, oh, I see it.
Okay. So I'm in this section where we're defining a trait. So all you need to do, you can just continually, so when we do SP $, we're just continually adding parameters to our SP object. So we could just do add trait A again. This will add a second trait. And let's see, I'll make a slightly different. We'll do. And then I'll go ahead and define my heritability. Oh, we need to, okay, so this is, I just ran into an error, and this is an important thing to note. If you have two traits, you obviously need to set two heritabilities. And we will do it like this. Heritability equals C, that means concatenate, I believe. So you need to feed it two values if you're going to have two traits. That should work now. Yeah. So the first value corresponds to our first trait. Second value corresponds to our second trait. And then let's see. That's not what I wanted to do. When you are making your selections, you can define an argument for which traits you want to select on. So that would be like, here, we're selecting families. I believe you can do trait equals. You want to define trait 1 and trait 2. This is not something I have personally a lot of experience with. But in general, what you're going to need to do is define all your traits in your sim params first here. And then when it comes time to make selections, you'll just add the traits you want to select on as an argument to this command. And that's kind of all the detail that I'm familiar with. I'm sure there's probably a bit more detail that you can find in the documentation. I'm not sure if there's any other questions, but I'll mention that there's a 7th script here, just like a sandbox to play around with the commands. So please feel free here to try on your own. I gave a general outline of what you need to do: create a founder pop, define your sim params, and then outline your strategy. So this might be a good time if you have some ideas you want to try them out here, and you can ask questions if you run into any errors.
So what are some ideas that people can, for example, play around with just to get some thinking started?
If there's anybody who's working on with cattle, wheat, or maize, you can utilize the fact that AlphaSimR contains predefined parameter sets for those. If not, you can create a new population and maybe outline the selection steps in your particular breeding program. Like maybe you don't use pedigree and there's another selection strategy that you'd like to model. You could start by just typing out the steps for each generation and then using the AlphaSim commands, kind of just translate it into AlphaSim terms. And what I'll do is I'll share the documentation with everybody. And you'll be able to see kind of a glossary of all the commands.
And they have the Highlander lab. They have YouTube training workshops too, right?
That's right. I will share that on edX of the SimR. Yes, there's a course called Breeding Programs Modeling with AlphaSimR.
So, for example, as you write yours, what are some of the crops that you're dealing with? Maybe you can type them in the chat and we can get an idea. There's a question here from Fabian. For hybrid crops, can it simulate different hybrid testing scenarios or mating schemes and changing number of testers, or changing number of testers? Yeah.
Yeah, I can show you how to define a custom mating scheme. I'll just need a second. To pull that up. So this is something I was playing around with recently. So yeah, I can show you this in the screen share. I'm gonna just, so I'm in script 7, my own script 7, my blank script. Yeah, so if anybody wants to create custom crosses, all you need to do is kind of create a crossing matrix. So here we have a matrix with 20 rows and 2 columns because these are 20 different crosses that I wanted to make. And the AlphaSimR command you're going to want to use is make cross. And you can indicate which generation you're crossing. And then the key here is the cross plan argument. So I have cross plan equals cross, which is the matrix I defined. And if you need, for example, to select particular individuals, all you'd have to do, like, let's say you wanted to pull the top individuals, you could pull the phenotypes of all your individuals and then order them in a ascending order. So you have like the top individuals at the top of your matrix or data frame, and then using those top individuals, like make the crosses you want to make. And I'm not an expert on making particular crosses. This is something that I personally haven't investigated a ton, but it is doable. And this is the general format for doing that.
So what are some of the crops that people are dealing with? Are we mainly talking at the attendance? Are you mainly dealing with clonal or line-based development of lines, or what sort of crops? So it's got Juan works on cassava.
Nice.
So
Thanks so
cassava is pretty much clonal, just like potato, right? So you produce an F1, and then that F1 is you produce all the various. From there, you just test the number of clones. Potatoes, sorghum. So okay, we've got a few hybrid-based crops. Eucalyptus. Is this camelina? Is it camelina? And acacia. Hybrid. Okay, working on hybrid breeding. Okay, so that you needed some simulation. Eucalyptus and pine. Okay. So we've got a variety of types of breeding programs here. So some hybrid-based breeding programs, and others line where inbred line development programs where the line is the variety. Another ones they involve in developing inbred lines, but a hybrid is the variety. And then others that are clonal. So what are Isabella? What are some terms that people probably need to be, some key terms when they're building up their steps that people need to be aware of?
For the clonal species, there is a framework for modeling that in AlphaSim. And it looks like I've not done that myself, but it looks like when you are setting up your simulation parameters, you do simparam $clone, like in that area where we add all the traits, you can add the information that you're trying to do cloning at that stage. Let's see. Yeah, so my suggestion would be to think about what the parameters of the population that you work with, and start by trying to model that founder population as accurately as possible. And then again, think about the traits that are most important for you and what those traits are structured like. What is the genetic architecture that you're trying to model? And then you can use the simparam feature to create the environment that will simulate as closely as possible to reality what you are trying to model with your crops.
So in cattle, so somebody works in cattle. So in the case of cattle, you're doing testing, progeny testing, right? So you're essentially, unless you're using genomic selection. So let's say you're not, would there be a way to incorporate progeny testing into the strategy? So that would be, you make a cross, and then based on the progeny performance, then you select the best male. So I think that's probably codable, right?
Yeah, for folks who are working with animals, when you are defining your population, you have the option to define male and female. So I think that would allow you to do that sort of testing that you need to do for those particular progeny. Just like a more realistic modeling of crossing between males and females in cattle. But I personally have not done that because I work with beans, but there is a framework for.
Yes, they do have a whole aspect or part of the AlphaSim suite that's for animal breeding. Yes. Okay. So we dealt with clones, wheat and sorghum. You're developing inbred lines, but then you can later on say cross those inbred lines and test for superior height, and then you could extract estimates of, say, general combined with the raw data coming out of AlphaSim, you could then extract estimates of general and specific combining ability for your hybrids, and that's one way that you could also simulate, which avenues or pathways might lead you into the superior combinations, or perhaps even simulating heterotic groups if you had that sort of question. So okay, and then eucalyptus. I'm not really familiar with not very familiar with tree breeding. I do know it takes very long, but my sense is that it's also line development. Somebody correct me. Or is it clones primarily, say in the case of eucalyptus? Yeah, okay. Yeah, I think with say pine, I believe it's clones. So okay, so you could simulate that sort of the same way as you would say potato or sweet or cassava. One question, Isabella. So for example, if people had SNP data, right? So if you had the SNP array data, okay, so is there a limitation to the size of the matrices that you can input into AlphaSimR?
Not that I'm aware of. The key is that you have data on which chromosome each SNP falls. Without that bit of information, I don't think it's possible, because AlphaSimR will model on a per chromosome basis. So yeah, it's just a matter of creating a separate object for each chromosome, and then you can go ahead and with your simulations for your actual population.
And you can use, say, linkage maps versus physical maps. It doesn't matter.
I believe that's correct. Yes. Yeah.
Wonderful. Well, we'll have a minute here if for wrap up. If anybody has any feedback on the quality of the workshop, or any sort of comments, or any further questions at this point, but mostly if you have comments about what would you like to have seen in this workshop, that would be very much appreciated. If you have those types of comments, please type them in the chat at this point. We'll give you a minute to do that. But given that it seems that everybody was everything went very smoothly, so it's a great tool, right? Posit Cloud. It enables you to have your own, and you have your own code, and you can play with it without having to depend on a number of packages or files that would have to be sent around via email and things like that to load them up into R. So it's really a great tool for teaching.
Yeah, thank you everyone for your participation, and please, any comments, we're happy to hear them.
Wonderful. Well, with that, thank you, Isabella, for preparing this for us, and for having such a detailed and clear workshop. I think it's wonderful to see that people were able to follow it, and it seems like there was not a lot of engagement and interest. So thank you, everybody, for attending. And so we will upload this, we'll edit it, and we will upload this recording to YouTube if you want to go back. And we will take the email list for the registrants of this workshop, and we'll send you a few of the links and resources that were included. All right, so you should have access to your own copy of the R Cloud code, so that you can play with it as much as you like. There are other resources, but as far as documentation, we'll send that out. Do you have anything to add, Isabella?
I don't think so. Thank you, everybody.
Great. See you, everybody. Take care.